Okay, thank you very much for the introduction. This is my first time at CLINAM and hopefully not the last one. I took this opportunity rather to discuss what I think are the challenges in dendrimer chemistry so they can be applied to nanomedicine rather than to present my work that I have been doing in my career. But I will be open to discuss any of my work if you're interested. I have been working with Dendrimers for 14 years already, since my undergrad. And I will talk about this landmark paper that was by Tomalia in the 90s. And it has been 25, 26 years since this paper. And in this article, Professor Tomalia not only coins the name Dendrimer, but also gives a vision what the Dendrimer field would be in the future. And extracting some of the words that Dendrimers could be used as platforms to build a wide variety of materials, including drug delivery prototypes and synthetic vaccines. And I think this was a vision where, at this point, Dendrimers were still being developed. So just in the beginning, I think to join Professor Tomalia, there were other big names that are worth mentioning. Professor Majoral, of course, with his beautiful, intriguing phosphorus dendrimers. I think Professor Didier Astruc, who I was very happy to work with, Professor Frichet, and UNICAMP, I think are probably, in my opinion, the pioneers in this field. And since the 90s, there was a lot of groups working on dendrimers, not only on optimizing their structure, but also on trying to figure out different kind of applications. And in terms of nanomedicine applications, dendrimers have potential in drug delivery, theranostics. We heard today about photodynamic therapy and for a variety of diseases. And of course, today was very interesting to hear about the importance of inflammation, which I think was very, very important for me to listen about that. And I hope everybody learned as much as I did about that. So why I think people are very intrigued by dendrimers is because I think this is the only kind of nanomaterial that we actually have a precise control over their structure at the molecular level. And that allow us to put to control exactly their size, their structure, the type of functionalities we have on the outside, on the inside, and we can put a precise number of functionalities on the surface, on the core, at the branching points, or even encapsulated between the branches. But one of the major, not the major, but one thing that has been focused on is the possibility of introducing multiple functionalities in one dendrimer. I think in this session we heard several talks more turned to the dendrimers with one functionality, I mean multiple functionalities, but all equal, I mean homofunctional dendrimers. And what I want to discuss with you is the multifunctional dendrimers. So there is more than 20,000 articles in about dendrimer science, and we can hear from several people that are not within the dendrimer community that sometimes when I look for collaborators, they say, oh, dendrimers, they've been around for a while and they don't go through, the clinical trials are not working, and that's very annoying in my point of view. I don't know if you have that experience. So it seems like the number of dendrimer-based products that are advancing to clinical studies has been a little bit deceiving. And especially I'm talking about the multifunctional dendrimers. And why do you think, and I would like to hear your opinion, why you think these dendrimer-based products are failing at passing the clinical studies? And what we hear about the multifunctional dendrimers, and I'm talking about the formulations that are trying to make dendrimers with targeting groups and drugs and imaging agents and solubilizing groups all in the same molecule. So what is happening is that there is a lot of batch-to-batch variations. There is lack of reproducibility on the pharmacokinetic behavior, poor characterization, and a lot of impure compounds. And this is intriguing because I just told about how dendrimers are supposed to be these perfect molecules, right? So what is going on? So this is what I think it's going on. When we go on the literature, there is thousands of articles that actually use this kind of approach, which I call random statistical approach. So because only homofunctional, I say monofunctional, but I think it's more correct, homofunctional dendrimers are commercially available. People will add multiple functionalities using this random statistical approach. So they take, and I'm not even talking about what the quality of the PAMAM they're buying that Professor Tomali already talked in his previous talk, but let's suppose you're buying the perfect PAMAM dendrimers from nanosyntons, right? Even then, what people mostly do is you have all amine groups, and I'm going to bother you a little bit with chemistry, I'm sorry about that. And then you will add, let's suppose you want 3 functionalities in your dendrimer. So people usually add about 1/3 of the equivalence of the amine groups, and then they will have about 1/3 of the surface functionality with this. Let's suppose this is a drug. And then they will add a new reaction with about 1/3 of the equivalence of the remaining amines, and they will have this bifunctional dendrimer. In this case, let's say it's a drug and an imaging agent. And to finish, let's suppose this is a targeting group. You add enough to functionalize the remaining amines. So at the end, this is what we expect to have. Multifunctional dendrimer with about the same amount of drug, targeting groups, and imaging agent. This is the expectation. This is what really happens, okay? So when we do the first reaction, we can have from non-functionalized dendrimers to totally functionalized dendrimers to countless types of combinations. And this effect is propagated and amplified in the next steps. So what we have at the end actually is a complex mixture of species that are very difficult to identify and to separate. And I'm not inventing this. I'm just giving some examples of characterization that I can find in literature. And sometimes as a reviewer, I don't really like this kind of spectra. And sometimes people don't even put their spectra, just numbers. This is how a Malditov can be of this species. So you'll have a mass between, I don't know, 15,000 to 30,000. And then people will say, oh, my mass is 33,849, you know. And this is not very good in my point of view. And also, when you characterize by NMR, if you can actually integrate these peaks, you will have an average of the groups you have in solution, but you don't have any information on the individual species, which dendrimers have how many, you know. Okay. And actually, there is a very nice paper from the Baker group where they analyzed a dendrimer that they had previously published as they published that their dendrimer had 4 folic acids and 5 MTX drug molecules. And so they took that molecule they published and they reanalyzed with HPLC, high resolution, and did some complicated peak fitting analysis. And what they got is that their product that was published as being in average 4 folic acids and 5 MTX drugs. Actually, they had about 182 different dendrimers in solution, you know. So only about 4% of the dendrimer sample is composed of the expected dendrimer. So we have been hearing in this conference a lot from FDA people, and we don't need to actually get into discussion how 4% actually represents, and 182 different species will not pass regulatory revisions. So putting back this slide, so I think, in my opinion, that when we make these multifunctional dendrimers through this random statistical approach, this loss of control over the final structures actually contradicts the purpose of using a dendrimer, a perfect molecule, let's say. So we could see here all the work when Professor Majoral presented on the preoccupation on showing how many functionalities they have, and Professor Tomali was discussing how important it is to have specific number of functionalities and have a very well-defined structure. So this is, I think, is one of the main challenges right now in dendrimer chemistry. So I believe that the future of dendrimers application in nanomedicine depends on batch-to-batch reproducibility of the final materials, and I believe that to advance these dendrimer-based materials to clinical trials, it is very important to actually develop synthetic strategies to build well-defined multifunctional dendrimers, where we have a very controlled number of how many functionalities of each group we want to put. So I'm out of time, but I have more slides, if that's okay. Okay. So just in literature, we can find different types of bifunctional dendrimers. So we actually know how to make bifunctional dendrimers with a defined structure. We can have functionalities on the core, on the outside, genus dendrimers, dendrimers based on bifunctional dendrons. Just some examples. This is a phosphorus dendrimers from the group Professor Majoral, where he selectively introduces two functionalities. Genus dendrimers, where you can actually have PAMAM and polyester free-shade type dendrimers joined together through click chemistry. UNICAMP type of dendrimers with two functionalities. So we can do bifunctional dendrimers it's okay. Three-functional or more functionality is more challenging. There are a few examples in literature. Triazine dendrimers is a good example, or fullerene-based is also a good example. And this is what I'm working on right now. I'm trying to develop synthetic strategies to build well-defined multifunctional dendrimers. So we use actually attach different dendrons to a trifunctional core, or use symmetrical dendrimers and then attach bifunctional or trifunctional dendrons, or combine genus dendrimers with bifunctional dendrons. So just very quickly, this is one example where we have a trifunctional core that we can actually selectively add different dendrons, and we have dendrimer with a specific number of specific functionalities. This is a different approach we are still working on. Have some combination between genus and then multifunctional dendrons. This is an example that we successfully synthesized. It's a dendrimer that has 54 acid groups, 9 azide groups and 9 dyes. These groups are available to attach either more functionalities or attach the drugs and their targeting groups if that's what we want. This is a new dendrimer we're working on right now. I'm not going to talk about the application or which drug or which application, which disease I'm trying to do. Actually, what I want to focus the discussion is on the platform. So why I think it's time to meet the expectations, because even with all the drawback associated with the random statistical functionalizations, Dendrimers have showed that they can be very promising for nanomedicine applications. And some trends related to size, branch structure, surface functionalization are being unveiled. And there are new synthetic tools available. We have a new pool of reactions of click chemistry, orthogonal chemistry, so we can actually make these complicated molecules, but with a very precise structure. And again, I think dendrimers are the only nanomaterials with this precise control over the final structure, and we need to be very careful with that and not just make complex mixtures out of it. And I think nanomedicine needs dendrimers. There are a lot of new therapeutic solutions that need to be resolved. I'm going to go faster. There is this outdated perception, in my opinion, that dendrimers are complex, slow, tedious synthesis, and very expensive. In my opinion, the very low amount of dendrimers that we will use actually will dilute this cost. And platinum is not cheap, gold is not cheap, right? So Taxol is not cheap. So I don't think the cost would be a problem, shouldn't be a problem in the first place. So it might happen that the well-defined multifunctional dendrimers fail in the clinical studies. It might happen, like many other promising drug formulations, but I don't think they should fail due to lack of reproducibility or impurities. I think that shouldn't be the case. And I strongly believe that dendrimers will, I think it's the time that we will make them a real product, and we saw so many nice examples today of applications of dendrimers, and I think it's a very exciting time to be a scientist. And I had a few more slides, but I will just go further. This would be the long-term goal, just to build a library of dendrons, targeting groups, and dyes, and just make a platform where we could just build a personalized dendrimer for each application. That's a very long-term goal. So, yeah, I want to thank you very much for the invitation, and my funding agencies, and all of you for your attention.
As a young generation perspective, I think you captured some of the critical problems, and I think your approach of modularizing with well-defined chemistry to bring these pieces together, rather than using stochastic, randomly attached conjugation chemistry, which really doesn't work that well. You're correct. But there are ways around it, and I think Star Pharma has mastered many of those approaches to have gotten as far as they have. And so we thank you very much for being so perceptive and telling us your view from the younger generation. She's one of the newer Dendrimer chemists, as opposed to people like Jean-Pierre. I won't mention anybody else because they're all much younger. But I would like to thank everybody for joining us this afternoon. Any question? Maybe one quick question.
Could I make one plea as the people who are not chemists, but at the other end of trying to drive this into the clinic? And I completely appreciate the point you make about the fact that we almost certainly have mixtures at the end. You're right, we shouldn't have impurities, but we almost certainly have mixtures, and you showed some nice pictures of that. and those isomers will be almost impossible to separate or even to identify. And I see Jean-Pierre is shaking his head vigorously, because we've all had the pain of spending years, and I've gone grey on it. I had no grey hairs before I started trying to sort out the mixtures, because you can't. I would ask you to bear two things in mind. First of all, Roche and Schering-Plough made pegylated interferon for hepatitis C, for which they made billions. And if you dig very deep into their patents, you find out that they were mixtures of isomers, because they used non-specific conjugation chemistry, and there were something like 30 isomers present. But what happened is that because they got them to the clinic and they made so much money, then they went back and reinvested the money, hundreds of millions, in analytical chemistry to make much cleaner preparations. The other thing that's very important is if you look at antibody drug conjugates, which is the most sexy thing happening in cancer therapy now, you find that when Genentech made their original antibody drug conjugates, and remember you have fab fragments, so you have four ends on which you can put the drug. They had mixtures of 1, 2, 3, and 4, and they then put those into the patients as mixtures, and I wished Anno was here, but he's gone. They put them into patients and they worked. And then if you dig very deep, you find Genentech wrote several patents in which they spent a huge amount of money doing the mass spectrometry required to go from 25%, 25%, 25%, 25% to where they are today. And do you know where they are today? They have 80% or 85% of one species, but they still have 15% of the other species. So we can either go back to chemistry, and if you go back to chemistry, and as you take over from the old men here, you will spend your whole life getting very little money and making a perfect product. But by the time you make the perfect product, nobody in the clinic will be interested. But if you say that we have a product that is good enough to justify testing in a very serious illness, and if we see some efficacy, big companies will come in and do all that analytical chemistry for you and solve the problems for which we don't have the money.
Those are excellent comments.
That's a very good point of view.
I'll add to this. I work, my group works with two major U.S. pharmaceutical companies, and we are using some of these modular approaches here. They have no concerns. They believe these will easily get into clinic. And so they are already analyzing the problem, and they don't go to the old-fashioned stochastic, oh, I'm going to put 4 different groups, very conveniently on every dendrimer.
1 second. I would like to add on the comment of Sunil. I fully agree. Sorry.
Yes.
I fully agree with Sunil's comment. That's why we need one clinical trial successful. That's it.
That's right. Hopefully you have it coming. Any other questions here to this young lady? To Professor? Yeah.
Yes, I just want to comment. I agree with you with the polydispersity of Dendrimers. And that's the reason several groups are working how to avoid these discrepancies. And I think you was mentioning the use of bisdendron. This is a good answer. There is another thing. People are working more and more on the synthesis what we call onion peel dendritic structure. That means that each layer is different from the previous one and for the next one. And each layer, with each layer, you can do a precise reaction. That is to say, for example, you can put a drug at the generation 2 and you put a drug on the surface. And there is a group of René Roy in Canada working intensively in this field, and we published recently in chemistry the first onion peel dendritic structure. So this is a way, because having different layers, you can do precisely the reaction you want to do at the given position, close to the core, close to the surface, or really within the cascade structure. And so, to my opinion, to avoid this problem of polydispersity, this might be the way to work, to and we are on the way to working in this field, and some other groups also.
Thank you, Jean-Pierre. If there are no other profound questions, so we give you a few minutes before the next session. Please join me in thanking Catia for her excellent perspective to wrap up this conference, and also to thank all of the fantastic speakers. I learned so much today. And I just want to thank all of you speakers for accepting our invitation and joining us at this fantastic CLINAM meeting. And if you want some good luck, just rub the CLINAM dwarf here so we can solve all these problems if the science doesn't work as well for you. So thank you very much for coming.
Thank you.